honor to be the first speaker of this session, and I hope my presentation will pop up on the screen soon. Okay, so today I'm going to talk about an option to cryopreserve larger volumes human semen, which means above 20 microliters, without using any permeable or non-permeable cryoprotectants or any freezing media. So, as you probably know, the male factor infertility is responsible for about 50% of the reproductive problems of all couples worldwide, and that's why the invention of facile, inexpensive and harmless method for cryopreservation of human ejaculate is a global societal challenge. So within the last 50 years in the field of cryobiology, there is one obstacle that is persisting, namely if we freeze the suspension, the cellular suspension very slow, then the cells undergo rheological deformation due to the fact that they become surrounded by liquid-filled ice channels that progressively decrease in their size and just smash the cells. Or on the other option is that the increased solute concentration causes osmotic shock. On the other side, if we freeze the suspension very fast, then there is no time for the intracellular water to leak out, which causes detrimental intracellular icing. And as a secondary effect, due to the diffusion-limited ice crystallization in the extracellular compartment, the warming is accompanied by fast water influx in the cell and its burst. So based on these two opposite on the cooling rate events, the current success rate of the artificial insemination from the first attempt is about 17%, with the intracytoplasmic sperm injection is between 20 to 30%, and the success rate of the classic IVF is about 10 to 45%, which means that, for example, on every 100 couples, only 45 will be able to have children from the first attempt. So what we can do? The classic approaches are to use permeable cryoprotectants. These are chemicals that permeate the cell membrane and distribute equally in the intracellular and extracellular space, but and increase the hydraulic conductivity of the suspension. But during the equilibration stage, they pass the cell membrane with velocity different than that of water, which causes multiple shrinkage and expansion of the cells and their mechanical deformation. This is the toxic effect of the permeable cryoprotectants. On the other hand, the non-permeable cryoprotectants locate only in the extracellular space of the cells, and they increase the osmolarity and also the dehydration ability of the cells. But according to some literature data, these cryoprotectants are also a little bit toxic. The third option, mainly developed by the group of Professor Vladimir Isachenko, is to use preservation media without any cryoprotectants. But in this case, you need to select the best, the spermatozoa with the best motility by density gradient centrifugation, which again, according to literature, sometimes causes DNA fragmentation. So how we can overcome these problems? We offer to overcome these problems by modifying the inner walls of the freezing containers by making them extremely water repellent. So such modification makes several things. First, the solid liquid contact area is replaced by solid air liquid contact area. And also the cohesion forces in the liquid becomes dominating over the surface adhesion forces, which force the liquid to acquire a spherical shape and reduces the contact with the solid surface. So from that moment on, by physical inherent means, the heat transfer rate and the overall heat transfer per unit time is decreased. So you can basically reduce the amount of ice forming at any sub-zero temperature, and hence regulating the buildup of solutes and the size of liquid-filled ice channels. Using this technology, by slow freezing and slow warming at room temperature, we managed to achieve 100% survival rate of the human spermatozoa. Not in too many of the patients, but from nine tested patients, we managed to do this with 2 of them. And also we noticed that this result is achievable even if you use different cryoprotective mixtures without paying attention of the particular cryoprotectant. These results are published in 2 papers in Cryobiology Journal in 2024 and this year. So based on these results, we decided to try to cryopreserve human semen without using any cryoprotectants. How we did that? By 2-step freezing. We fabricated sheet metal cryo boxes and coated their inner walls with super hydrophobic soot. Then we placed 50 microliter droplet in this cryo box, immersed its bottom and inner walls in liquid nitrogen, which caused initial first solidification within maybe 2 to 4 seconds. Then the sample was moved in a container with liquid nitrogen, where it was exposed to the vapor of the liquid nitrogen for about 1 hour. Then we removed the sample and slowly thaw it at room temperature. We did, to make a comparison, we did some control experiments with standard cryo vials and cryo straws, and also with in a culture medium without any cryoprotectant, and in centrifuged seminal plasma containing spermatozoa. And you will see that with the straw, the maximum survival of the motile species is about 1.6%, with the vial is up to 0.8%, while with the soot-coated cryo box it reaches up about 6%. So what is the mechanism behind this result? You can see on these 2 photos the different colors of the semen. This is during the first stage, during the immersion of the cryo box in the liquid nitrogen, and you see that the droplet has, let's say, gray-like color, and after the full preservation in 1 hour in liquid nitrogen vapor, the color changes to milky-like color. So we believe that here the semen is put in a stage where a lot of ice nuclei form in its bulk, but without any ice formation, and this justifies the gray-like color, whereas in the second phase these crystals form, but much slower, so the intracellular water can leak out without causing the damage. And the other thing is that the freezing, as you can see here, proceeds without the formation of a singular tip. This singular tip in watery liquids forms due to the volumetric expansion of the water and also due to surface tension effects. So if you don't have such a tip, this means that during the freezing, the mass density ratio of the water and the ice inside the droplet is relatively equal. And the third evidence is you can see here on the cryo box when we take it out and expose it to room temperature, some parts where the droplets occupy the cryo box don't have frost on them, which means that the seminal plasma itself, you know that it contains some sugars and amino acids, so probably these are working as natural cryoprotective agents and help also to achieve motility even without the use of artificial cryoprotectants. And in the case of centrifuged seminal plasma, this result is not observed, probably because the centrifugation breaks some of these biomolecular complexes. And so to visualize what I was telling you, I will show you 2 videos. On the first video, you see the first ejaculate before cryopreservation. This is how it looks. And on the second video, you will see the result after the instant freezing without any cryoprotectants by using the soot-coated cryo boxes. You see that we still have plenty of forward-moving species. So if I can summarize what are the benefits of this technology, firstly, it is extremely inexpensive because we generate the soot from the incomplete combustion of rapeseed oil, which 1 liter costs about €5, which is enough to process hundreds of cryo boxes. And the cryo boxes themselves are produced by tin cans. So we go into the so-called circular economy. We use the waste into useful product. The second thing is that the freezing process is not limited to small volumes. We managed to freeze 50 microliter volumes, and now we strive to freeze even higher volumes because we believe that the sperm volume also affects the outcome, but because the freezing is stochastic process, so no one says that the ice crystals form simultaneously. Probably they form gradually, and big volumes make the distribution of these ice crystals very sparse. So during the warming stage, there is no recrystallization, which can be detrimental to the cells. And also, the method is applicable to any patients regardless of their reproductive health status. And despite the parameters of fresh ejaculate, this technology offers survival between 250,000 and 5,500,000 progressively motile spermatozoa per 1 milliliter of ejaculate, which means that the method can be used in all cases for classic in vitro, for artificial insemination, and for ICSI. And also there is no need of qualified, highly qualified personnel, because you take the ejaculate, liquefy it, and without any processing, you just freeze it, thaw it, and you have a live species. So that's it. Thank you very much for your attention. If you have any questions, I will be happy to answer.
Thank you very much. I will just open the floor for questions.
Very good presentation. I have one issue because the motility for me is very low. Have you analyzed the DNA fragmentation and the proportion of viable sperm? Because maybe for ICSI maybe it's useful, but for in vitro fertilization with this motility, maybe do you need to sperm selection previously to I don't know.
Very good question. Actually, this is one of the reasons probably that my short communication was less rejected by the editor-in-chief of cryobiology, because she asked the same, you present only motility and you don't talk about DNA fragmentation and ROS production, but the truth is that we don't have, at the moment at least, the facilities to see if the DNA is damaged and if the reactive oxygen species are produced. And the other thing is, I agree, the motility, the success rate of this at the moment is low, but these are pilot results, first of its kind. We need to optimize the process. Maybe we're going to achieve much better motility. And in fact, as I said in the beginning, by using permeable cryoprotectants in 1:1 volume ratio, we manage to achieve sometimes even 100% success rate. So probably if we use the less harmful non-permeable cryoprotectants, we will be able to have maybe like 70 or 80% success rate. I don't know yet. This will be part of another project, because this project, it's already ended, and I just extend the deadline to be able to present the results here.
Okay. Any other questions? We still have room for another one.
Technically, what's the problem with using cryoprotectant?
No, wait. Otherwise, the people
I will tell you.
hear you.
When we use these commercial cryoprotectants, even at room temperature, depending on probably on the biochemistry of the seminal plasma of some patients, with some patients the decline in sperm motility is about 10%, but with some other it's about 30%. Especially if you use DMSO, it kills them. It kills them. I saw this with my eyes.
But does anyone use DMSO for human sperm cryopreservation?
Pardon me?
Does anyone use DMSO for human sperm?
I think I saw in the literature that they use, but probably in lower concentration. I used in 5% concentration.
Thank you.
Actually, I want to clarify, the reason we use DMSO is not because it's standard, for example, but because we wanted to examine how different cryoprotective mixtures will affect the outcome. Which one will be better? So we use several commercial cryoprotectants and also the DMSO in mixtures with different volume proportions.
Thank you.
Just one last question before, and I was wondering, maybe you said it and I didn't get it. What was the composition? When you were using no cryoprotectants, so what was the composition of the external? It was just seminal liquid?
Yes. Ejaculate, liquefied in a plastic container, sterile plastic container, and then you just put it in the cryo box and freeze it. That's it. That's why I'm saying that this technique is very promising, because you don't need to process the semen, you don't need to have medical stuff to make gradient density centrifugation or swim up or whatever. You just take it, put it, take it out, we have a live spermatozoa.
It's coming back to the gentleman's comment about if you are going to do an ICSI procedure at the end, and if it's not sufficient enough to perform IVF, then what's the point of really cryopreserving this way.
Right.
Because in many human IVF clinics, essentially the freezing protocol is very suboptimal because they don't want to use
I agree.
IVF.
I agree, but
But it's extremely important that you actually test the DNA fragmentation.
I will once
If-
we get the facility.
Exactly. If you get lower DNA fragmentation, then it will be an alternative.
Okay. I want to tell you that with these two videos that I show you, the overall motility after the cryoprotectant-free cryopreservation is about 6%, which means 5,500,000 spermatozoa. Believe me, if they are not DNA damaged, this means they are enough for classic in vitro, because for classic in vitro you use like 100,000, 300,000, up to 1 million, and we have 5 times more.
But assuming that you don't get any lower fertility after the separation or enrichment.
Yes. Okay, yes, yes.
Thank you.
Well, let's just give a round of applause to our speaker.